as we honor Oliver's memory, it is my pleasure to share more about his academic journey and the life he led with such distinction. After completing his undergraduate degree in physics at the Technical University of Karlsruhe in Germany in 1990, Oliver pursued a PhD at ETH Zurich in Switzerland, finishing in 1994. His studies of microelectromechanical systems, or MEMS, during his doctoral work laid the foundation for his independent career. Joining the Georgia Tech faculty in January of 2003, Oliver's contributions were vast and varied. His work, especially in MEMS and CMOS-based microsystems, is widely respected in the community, with over 190 publications to his name. Oliver served as the executive director of IEN, one of Georgia Tech's 9 interdisciplinary research institutes. In this role, he united Georgia Tech researchers and technical staff to support infrastructure for and advance interdisciplinary activities in the areas of electronics and nanotechnology. IEN boasts nearly 30,000 square feet of cleanroom space and hundreds of pieces of equipment for cutting-edge research in nanotechnology. IEN's over 200 faculty member affiliates and nearly 50 full-time staff make it one of the largest research institutes at Georgia Tech. Oliver also directed the coordinating office for the National Nanotechnology Coordinated Infrastructure, the NNCI. The NNCI is a network funded by the National Science Foundation that provides researchers from academia, small and large companies, and government with access to these leading-edge nanotechnology facilities. The NNCI's 16 sites encompass a wide array of capabilities and expertise in nanoscale science, engineering and technology, supporting and advancing research and innovation in the field. Oliver also served as director of our local NNCI site, the Southeastern Nanotechnology Infrastructure Corridor, or CNIC. Beyond his academic success, Oliver was a mentor, a colleague, and a friend. He was known for his sharp mind, gentle style, and unwavering support for those around him. His ability to foster collaboration has left an indelible mark on the community near and far. I am particularly pleased to note that a portrait of Oliver now adorns the atrium of this building, a fitting tribute to a person who dedicated their life to advancing our field through scientific excellence and community building. This portrait stands as a symbol of his enduring legacy. Today, we are also privileged to have with us Professor Andreas Hierlemann from the Department of Biosystems Science and Engineering at ETH Zurich. Andreas received his undergraduate and doctoral degrees in chemistry at the University of Tübingen in Germany. He held postdoc positions at Texas A&M and Sandia before returning to Europe to start a position at ETH Zurich. His research focuses on the development and application of microsensors, microfluidics, and microelectronic technologies with applications spanning systems biology, drug testing, personalized medicine, and neuroscience. Andreas's scientific contributions embody the innovative spirit and excellence that Oliver championed throughout his life. Following today's lecture, I invite you all to join us for a reception in the atrium. This will be a time to reflect on Oliver's contributions, view his portrait, and celebrate his life and legacy. So thank you for being here to commemorate Oliver. Let us carry forward his legacy of curiosity, collaboration, and compassion, inspiring future generations in the field he so dearly loved. Now, please join me in welcoming Andreas to Georgia Tech.
Thank you very much, Michael. Thank you very much for a kind invitation. And of course, it's an honor, and it goes without saying that I would follow this invitation to honor Oliver today. Let me maybe start with a couple of personal remarks before I go into the science. This is Oliver, myself, and Henry Baltus. Some of you may know him, when he received the Bosch Award 2016 at the MEMS in Shanghai. And Oliver and myself, we shared an office in Henry's lab, and I came there in 1999. Oliver was already there, and then we had 3 common years where before Oliver then defected to Georgia Tech. We also met privately. That was in Oliver's home, 2016 as well, in summer, before we then traveled on to the Hilton Head meeting that some of you may know. Let me say what impressed me most with Oliver. It was his innate friendliness. He was always supportive. He was always motivating students. I never heard a harsh word of him, only once, but that had very good reasons. It was a very resistant and deeply relaxed student that needed some motivation. And he had an extremely positive attitude to life, which I really learned to admire, because many things that were self or without problems for me were a daily fight for him. And he never complained, he never lamented. He was extremely positive character. And that is what I take, or what impressed me most and what I take from as his legacy. Now let's come to the science. So this presentation will be two parts. There will be essentially one part, which is the first part, mostly about microfluidics, hanging drop networks and microphysiological systems, and there will be a second part on high-density microelectrode arrays. In the first part, I give two examples for such systems. One is a microtissue-based system, so-called embryonic stem cell test, and the second is a barrier system, the blood-brain barrier, that we develop for drug testing, for essentially also studying basics of this kind of biological systems. In the high-density microelectrode area, I will speak mostly about neuronal systems, characterization of those, and how you can essentially handle and interact with neurons and what it is good for. The prediction dilemma in substance testing that we all know is, on the one hand, there's the 2D cultures, but they do not reflect or recapitulate the microenvironment. Means cells are not in tissue, they are in tissues in the body, they have an environment, they have chemical cues, and they are not essentially under themselves in a petri dish. And also there's organ interactions in the body. That means there's metabolic effects, there's a lot of interaction between the different organs which are not reflected in a 2D petri dish. On the other hand, there's animals, but there is interspecies differences. For example, rodents never get diabetes, as much sugar as you feed them, so you cannot use rodents as an example for studying diabetes. So what is a solution? Not the solution, but a solution to that problem, and we think one of them is advanced in vitro systems. This is the well-known drug development pipeline. You start out with about 10,000 compounds, and at the end, if you're lucky, you get one approved. And where do the systems, the microphysiological systems come in? They come in here, where you go from drug discovery, to the from drug discovery to the preclinical phase. The preclinical phase is usually the animal trials, and we try now to replace them more and more with human-based systems, means systems that use human cells of human origin. And therefore, we do not have interspecies differences anymore. And FDA approved last year, as you may know, for the first time that it is possible to go into clinical trials without animal trials in the meantime. So we can do that with advanced in vitro systems, and I hope that the ones I show you will, at least to some extent, demonstrate that this is possible. So what are microphysiological systems? Well, they are usually 3D cell assemblies, means they are microtissues or membrane structures as they also occur in the body, not only cells or single cells or cell cultures. They allow for comprehensive testing, and they allow for, studying tissue interactions, as you will see and as I hopefully will show you. So what is such a microphysiological system composed of? So on the one hand, organ models that you need, or tissue models. Then you need a fluidic system to connect and host those, and also supply nutrients, etc. And finally, you need also readout and sensors to show or to demonstrate the effects that the candidate drugs or compounds do have on your tissues. There is already, we are not the only one, there's already quite a collection out there. Most important or most famous probably the ones by Emulate, it's a company out of Harvard, Don Ingber's lab, and there is essentially also multi-organ chips like these from the Tissues company in Germany, or this one by Eddington et al. And then there have been other trials assembling essentially a lot of chips. You see that's a multi-chip solution with a lot of pumping and tubing in between to reflect interactions in the human body. So you can have one or multiple organs, preferably multiple, and you can recapitulate the organ physiology. That's the important thing here. So let's look at what we do, what kind of models do we use. we use so-called 3D spherical microtissues. And the production of those is very simple. You take a cell suspension, the cell sediment to the bottom, and you see that here, within 3 to 4 days, you get a microtissue. Of course, you have to have the cells that are relevant for the respective organ. For example, in the case of the liver, you have to have hepatocytes plus Kupffer cells plus other cells that are all in the liver. In other tissues, it's more simple because it's only one cell type like cardiomyocytes in the case of heart. You can do those, and we have a spin-off that produces those. You can even buy them for different organs, as you can see here. Most important for us is usually the heart, because the heart may not be damaged by any drug you test or compound. Then the liver is very important because it is a primary metabolic organ that metabolizes many of the compounds. And then there's others that may be target tissues for medication. Well, what we did is, that's the standard method to produce hanging drop systems. You have a hanging drop, isolated hanging drop, and then the cell sediment, and within 3 to 4 days, as seen, you get the tissue. What we now did is interconnect those hanging drops with channels. And that way you can place many of those in the same plate. For example, you can, with the same cell suspension, produce hundreds of microtissues at the same time in the same plate with very little scattering concerning the size and composition. Or you can essentially also, in different hanging drops, place different organs. That means you can place a cardiac tissue or lung tissue or, for example, a liver tissue in those drops and do a multi-testing at once, testing efficiency and also testing, for example, toxicity effects. The importance, of course, that those drops communicate. That means the liquid phase of those drops is connected so that whatever metabolic products, for example, can diffuse along this channel from one to the other compartment. The chip design is very simple. It's not really the rocket science as you see it here. It's a cut through in the length. So there's fairly large structures, about 3 millimeter in diameter. You have hydrophobic rims that essentially limit the spreading of the drop. And then you see the drops form underneath, and they are usually all at the same height, as you can guess. Fabrication is very simple, PDMS and or SU8 molding. So that's standard stuff in a clean room. Those are the essentially cuts through the other axes here, which are not so important here. The flow characteristics are very simple. So you have essentially the liquid coming in here and you pump it out there. You can also tilt the platform to activate the flow. And you see that they're always at the same level. This is simply physics. It's the Laplace equation. Drops with smaller radii pump liquid into drops with larger radii until all drops are equilibrated. So you always get essentially inherent equilibration and same volume in all the drops. Now I want to talk about embryonic stem cell test. What is that? Well, you take embryonic stem cells, typically from a mouse, and you then form tissues that are like little embryos, if you want to say so. Then you expose those tissues to a compound, whatever chemical has to be tested, and there's many that have to be tested. Nowadays, every chemical has to be tested on teratogenicity. That's what we test here, essentially whether they do damage to the fetus or the developing life. And then you have, after the exposure, you translate it to the next substrate where they spread out, and then you look with a microscope at whether you find a region where there is beating, cardiac beating. That means that the embryo developed, there is cardiac cells in there, and that you have essentially no major damage to your embryo through the chemical. What is the problem nowadays, or the pain with it? You see that you have to transfer it through 3 substrates. You first have a hanging drop platform. Next, you then immerse them into your chemical, and then you have to transfer to a third substrate where you have an adhesive coating so that they finally flatten out and you can observe the cardiac beating. Systemic effects are completely neglected. So you have only one, essentially only one chemical. Here, there is no other organ in there, no liver, for example, nothing in there, which is reflecting or recapitulating the situation in the human body. So that's one of the problems that is or that is with that simple test, that is essentially that you do not have, for example, a liver in there. If you have a compound that comes into a human body, there may be several things happening. For sure, the liver metabolizes the compound, and it may not alter its effectivity. It may make it completely effectless, or it may even make it toxic because it metabolizes the compound to something much more toxic than the compound itself. And of course, that's something that you need to know, because this embryonic stem cell test will show you a completely different result with the metabolic products than with the compound alone. And that's why we essentially developed that system here. You have a liver in the same liquid phase with embryonic body. You have fluidic connection where metabolites can be exchanged. And then you essentially, on the same substrate, you simply turn it around. Then you have a non-adhesive coating here so that the liver cannot flatten out while the embryonic body flattens out. And then you again detect the cardiac beating. So it's a much more simple platform. One single platform in which you do the whole testing, and you have the metabolic competence in the platform. That's how it looks like. So you have a hanging drop system, as I showed it you before. We have 3 non-adhesive compartments on each side, here and here, which are essentially hosting the liver tissues. Then we have 3 adhesive compartments in the center, where the embryonic bodies are hosted. And then we essentially apply some valving, which I don't want to go into detail now, to essentially fill those compartments and load them with the respective tissues. Then what we do is we have that system, as you see it here, tilting by a small angle of 2 degrees, and that 2 degrees essentially give you flow back and forth. So you have the flow through simple tilting, which makes it easy to put in an incubator, and you don't need a pump. And sorry, I have 2 thick fingers for that. You see here essentially the formation of the embryonic bodies. That's the first 5 days. Then you flip it around, you load the liver tissues, and you see that liver tissues do not flatten out during day 5 to 10, whereas the embryonic bodies or embryoid bodies in the middle, they flatten out, and you can then observe them. You see essentially a flattening on an hour scale, so that is fairly rapid. In the next slide, you see essentially a movie of the flattening out here of an embryoid body on an adhesive substrate. They immediately adhere to the surface as soon as they get to it. And then you see at some point it starts beating here, and that's essentially what you observe. And this beating is the proof that this embryoid body has not been harmed by the chemical to an extent that it wouldn't be alive anymore. So that would be a negative outcome of the embryonic stem cell test, if you want to say so. And that works. And what we now could show is, by using this combination of liver and embryoid body, this is the curve, toxicity, the curve in the ID50 curve without the liver. And you see concentration is on the order of, let's say, 2 millimole. If you now add the liver, you see that this concentration goes down drastically by a factor of 4, because the liver does what you see here. It essentially activates the compound, which is cyclophosphamide, it's a prodrug, and produces phosphoramide mustard, which interconnects the DNA strands, and then the cell dies. So it means reality is that if you ingest cyclophosphamide, it's 4 times more toxic than a simple embryonic toxicity test will show you, simply because there is metabolization to a highly toxic compound, which is phosphoramide mustard. So this system can essentially show you what the metabolism does, and it allows you to include metabolic effects in the drug testing. Another system I want to show is now a barrier system. TEER is transepithelial electric resistance. That's something to measure barrier tightness. Blood-brain barrier model is very simple. Here you see the natural blood-brain barrier of the blood vessel, lined with endothelial cells at the inside. Outside there are pericytes, on the brain side, and there is astrocytes. And you see that reproduced here. We have essentially the epithelial cells lining the liquid duct inside. Then we have here the pericytes, and then we have the astrocytes. And again, as you can guess from this arrangement, we do tilting, a slight tilting to get the blood flow. And there's a certain tilting strategy that allows you also to do almost unilateral blood flow. This is the platform. I don't want to go into detail of fabrication. It's not rocket science. Again, it's really rather trivial. Large structures of reservoirs, electrode generation, and PDMS interlayers, all glued together by double-sided adhesive tape. So there is not really much important technology in here. For the tear measurements, we optimized the electrode structures. We had the ring electrodes here around the opening. You see it. And we have essentially the ITO electrode on the other side. Why do we have an ITO electrode here? Simply because we want to come with high-resolution microscopy from the bottom. And we need a short focal length, and we need a transparent electrode for that. It's a 4-electrode measurement. As you can see it here, we used Zürich instruments, impedance amplifiers to look at the resistance values. Here you see essentially immunostaining images. That is the endothelial cells at the membrane. That's the membrane itself. That's the pericytes, the human pericytes and the human astrocytes. That's a close-up of an astrocyte in the focal plane. And that's the pericytes seen from the top. So you see we form a very tight barrier. You see here dynamic and static. Dynamic means we have a flow in the channel. Static means no flow. And you see that, of course, it gets more tight when you have flow, because this is what the cells are used to, blood flow. And then they orient and align and form a tighter barrier. And you see in monoculture and is less tight than the co-culture, which is also expected. The pericytes add to the tightness of such a barrier. And you see as the barrier grows, the tear value goes up, means the resistance, the electrical resistance through the whole system goes up, sorry. And you finally see here the experiments. That is essentially how it goes. You form the blood-brain barrier during 3 days by essentially seeding the cells and then turn it around. And then you do essentially the hypoxia treatment. And you see on the one hand the live imaging with high resolution through the microscope here, and on the other hand, the tear measurements upon a so-called ischemic insult, which means you simply do deprivation of glucose, that means the main nutrient, and oxygen on the other side. And therefore, of course, the cells do not feel happy anymore after being deprived of nutrition and oxygen, and they round up and the cell barrier loses its tightness. And you see that immediately, that the tightness of the barrier goes down like hell already after half an hour. And you see it also after about 60 minutes that the tight junction stains go away and essentially cells round up and change shape. So you can at the same time, with high resolution microscopy and electrical resistance measurements, observe the tightness of the barrier. Okay, let's switch gears here and go to the high density microelectrode arrays. They are made of CMOS on CMOS substrates. We work on those since now almost 20 years. And I want to present you the last generation system that we at the moment have, which is not yet handed over or transferred to our spin-off. That's a general situation that you may know. The electrogenic cells like neural cells or heart cells sitting directly on sensors, platinum electrodes usually. Of course, the chip is covered with laminin or polylysine to make the cells more comfortable. And the ions, which are going through the ion channels in and out, are moving charges. Moving charges produce voltages which you can detect. Here you see a typical patch clamp signal, which is a standard or gold standard method. And here you see the extracellular signal. What you see is, on the one hand, the polarity is a bit different or opposite. And on the other hand, you see also that there is 3 orders of magnitude, here microvolts and here millivolts in between. Means you have to do something about those signals. Why opposite polarity? Because the electrode in patch clamp is inside the cell. It means the sodium ions rush into the cell towards the electrode, whereas from the sensor outside the cell, they rush away. And therefore, you get a depletion of sodium ions, therefore negative signal. That's a neuron. I don't want to talk much about it. Information processes in directional. You have an input region, a dendritic arbor. Then everything is integrated here, and then it's decided whether or not to fire an action potential at the so-called AIS, axonal initial segment. And then this electrical signal runs down the axon and is handed over to the next neuron through the synapses. Why would you want to use microelectronics technology here? There's two good reasons. Number one is signal quality. If you miniaturize the electrodes, the signal levels get lower and lower, and it's more and more the danger rises that your signals get compromised by noise. So the closer you can get to the site of generation, and the more immediately you can amplify and filter signals, the better. And that's why we essentially integrate everything with the electrode itself near to the electrode. Second is connectivity. Those chips have between 30,000 and 60,000 electrodes on a 2x4 millimeter array, and there's simply no way that you can wire those out. There's way too many wires. You would need hundreds of thousands of wires to be able to do that, which makes no sense. There you can use essentially multiplexing schemes and other features that are available in modern electronics. Usability is for people that are not familiar with the system, so they can essentially simply with menus be guided through applying the system without knowing all the background. Of course, we are not the only ones doing that. There's a whole field of in vivo devices that have been initially developed by the University of Michigan, Ken Weiss and his group, and are now developed worldwide by a couple of groups. One of the most recent developments is the IMEC Leuven NeuroPixel 1 and 2, which is standard in many neuroscience labs nowadays. You see the NeuroPixel here. It's a long shank, which is about 10 millimeter long, 70 micron wide, and 20 micron thick. And you have 966 electrodes on the shank and all the recording channels on the base. And only 384 of those can be read at a time. NeuroPixel 2 has about 5,000 electrodes on 4 shanks and about 800 that are readable at a time. There's also the planar arrays, and they are fabricated and developed at Harvard, the group of Donny Ham, IMEC and Leuven. Columbia as well has some efforts on that, Ken Shepard's group, and then there's also in this field already companies out there, 3Brain in Switzerland, Multi-Channel Systems in Germany, and Maxwell Biosystems, which is a spin-off of our lab. You see essentially a chip of Donny Ham, and this is one of the open gate FET structures with a dielectric coating, means an oxide, aluminum oxide or silicon oxide, to ensure capacitive coupling. That was a notorious anxiety of Peter Frommer's at the time, that there would not be any electrochemistry effects, although there aren't any if you operate those metal electrodes in the right regime. So what are the readout schemes for those? You can either do the so-called active pixel approach, which is what you have in your CMOS cameras in your mobile phones. So you have a pixel in which all the circuits that are needed to read out and filter the signals of that pixel are jammed in. Of course, you make a trade-off here, signal quality versus spatial resolution. The smaller you make the pixel, the less quality of the circuits. So you can essentially, you have to make a trade-off which is useful for your application. Switch matrix MEAs is different because you have only switches inside the array and have all the circuits at the periphery. Means larger amplifiers, larger filter structures, etc. But you can only read out subsets of electrodes. But you can make larger arrays. You can make much more array electrodes, which you then have to scan through, of course. But the array can be larger. You see here essentially the principle of the two approaches, and you see also a signal on the same electrode on the one hand by an active pixel MEA, which is clearly more noisy than in the case of the switch matrix MEA. You see noise characteristics here typical is on a 10 microvolt RMS, whereas here we are on a 2 to 3 microvolt RMS, which is needed for cells or structures that are more or produce smaller signals. Axons, for example, you cannot see with 10 microvolt RMS. Also, iPSC-derived cells, which are usually very immature, have very low signal levels that are very difficult to recognize with high noise levels. That's the array that we developed. It has 60,000 electrodes. That's a density, 5,500 per square millimeter. And you have can read 2048 at a time. Every electrode is bidirectional, and you see also we have included additional units in here, which is impedance readout units, neurotransmitter detection, also low field potential readout, and current recording. So we have all those units to allow for maximum flexibility. And any of those units can at any time be wired to any electrode of the array. Of course, also multiple units at the same time to different electrodes, or multiple different functional units. That's the schematic. I do not want to go into much detail. That's the electrode array, the readout, neurotransmitter detection, impedance measurements, the digital control circuitry, controlling everything, and here the stimulation. I will go through a few details in very short time. Electrode array, we have a switch matrix readout, so it means 6 horizontal, 6 vertical lines. Each pixel includes 3 switches and the SRAM cells needed to operate those switches. Switches do, on the one hand, connect electrodes to array lines, and on the other hand, connect line to line. And that way you can essentially route almost any arbitrary configuration out in such an array. Max high density block is 45 by 45, which is almost the 2048 readout units that we have. Reconfiguration is quick. Pitch is 13.5 micrometer. Those are the action potential and field potential readout channels. Fully differential amplifiers, programmable in their amplification, depending on the preparation. For cardiac, you don't need much amplification. However, for neurons, and especially for iPSC-derived neurons, you need a full-scale amplification. Sampling at 20 kilohertz, and you see here also the ADCs. I didn't mention that, of course, we do not send out any analog signals off chip. It's all digitized on chip, and then bit streams are sent out because they cannot be compromised by noise or connection problems. That's the readout noise. You see the power spectral density plot here over the frequency band. That's the local field potential band. That's the action potential band. And you see the values here, which are decently low, so that you can get very small electrical activity detected. Stimulation units, we have current and voltage stimulation. You see the max values here, which are by a large margin enough to stimulate any electrogenic cell that could be on such a chip. We have a voltage limiter in case of current stimulations to avoid frying of cells through too high voltages. And we have an artifact removal, which I will show you. The stimulation artifact is a problem because you have millivolt stimulations and read at the same time microvolts. And you will see what we do about that. And again, you have ducts now to generate the stimulation pulse, which is almost any arbitrary waveform with 10-bit and 20 kilohertz. Cyclic voltammetry is also included, so that means you can do fast scan cyclic voltammetry on those electrodes. Of course, we do not want to use sensitive coatings because then we would lose the electrodes for the other functions. And this is the least explored unit so far, so we did not do much testing on that one, but we know that those features are all guaranteed, so we can go do ramps faster than 100 volts per second, and we essentially can do simple neurotransmitter detection. We prove, or we have seen that. Finally, the impedance measurement units also very important because you have essentially your array at the downside of any preparation, not the upside. With a microscope, you look on the upside, but you cannot see on the downside because the substrate is intransparent. So an impedance image is of great help to allocate electrodes and structures. So we have essentially lock-in amplifiers here, 32 on the array in 8 blocks of 4. And essentially the frequency range is between 1 Hertz and 1 megahertz, and the acquisition of an impedance image takes full frame or full resolution about 20 minutes. If you only want to get an overview picture, it's about 1 minute. That's now what you get from that. That's essentially an optical image. This is just as a guide of the eye. That's not the structure here. Where the prokinase cells are, you have the molecular layer, you have the white matter, which is myelinated area where there is no signals. You have the granular cell layer and the prokinase cell layer. Prokinase cells are the most electroactive ones here. You see those guys. And you see that immediately in the voltage recording that this is pretty much the Purkinje cells that are mostly firing that you immediately recognize. But the impedance images are also very useful. You see on the one hand the phase and the magnitude image. In the phase image you see mostly the adhesion of the slice to the surface, to the array, which should be as uniform as possible. And here you see really dielectric properties in the magnitude plot. You see here obviously the dielectric coefficient of this material is much lower than in the other regions. That is essentially these myelinated regions or white matters. You see pretty much the structures that you see here reflected and reproduced here. So this impedance image can help to show or to locate and characterize regions through different methods. Let's look at a couple of measurements. On the one hand, we can do all kind of preparations by simply mounting such a ring on it. You see what is the side view here, chip down there with laminin coating, and then you see the bond wires going out. Cells are seated directly on the chip. This is a false color electron microscopy image simply to show that the cells nicely and tightly adhere to the chip. That's essentially now a slice recording where you see typical activity starting at CA3 and propagating to CA1. So you get a high resolution, essentially electrical movie of the activity of a brain slice, a hippocampal slice in this case, with this characteristic structure. You also can look at cultured neurons and see here one neuron firing and how now the signal spreads and propagates in its axonal arbor. So that's only one neuron, that is after spike sorting, because if you do not spike sort, you get a lot of activity at the same time. If you do spike sorting, you can essentially allocate the electrode and the activities to a certain neuron by time allocation and by geographic allocation. That's the resolution that you can get. That's all electrodes. You see it here. That's the picture microscopy image of the respective neuron. The red staining is the so-called AIS, axonal initial segment, which is a region of highest ion channel density. And you see that the activity and signals that are recorded on those electrodes are completely different depending on which electrode you are. The activity always starts at the distal end of the AIS, electrode 3, and then on the one hand, back propagates towards the soma, you see it here, and into the dendritic tree, which is up here. And here it changes polarity, because here you have essentially positive, negative. That is simply due to the fact that a cell acts as an electrical dipole. So that means for any ion synced in the AIS, you have to have an ion source somewhere else in the neuron. And that is so-called return currents. So that means you get always positive first signals in the dendrites, whereas you get negative first in the AIS and the axon. And you see here that the other path is down the axon, which is those signals here, which get smaller and smaller because the structure of an axon is very fine. It is, in average, about 200 nanometer diameter. You also see that the footprint of such a neuron is much larger than its physical structure, which is not surprising because, of course, ions diffuse. That means you always have very highly redundant information. Now what can you do? And you can do all these characterizations at the same time with such an electrode array. You make a, let's say, 5-minute recording or 2-minute recording, you get all the information that I will show you in the next slides at once in the different frequency ranges. You can get the patterns of individual neurons, as you see it here. That's a still image of the same neuron where you see the axonal arbor. That's a latency map of this neuron as well. So you can assign components or compartments to single neurons. You can also look at subcellular compartment properties, like, for example, axonal conduction speeds. Or you can also look at networks. It means how does a network fire, how is network firing correlated with neuronal firing, how is a neuron connected in a network. All that you get at once. And I show you here a couple of examples. That's single neuron activity here that you spike sort. Here it's bursting, which is very typical for cultures. And then you can extract the single neuron traces and single neuron geographies or morphologies. What you get is typical firing rates and spike amplitudes and things like that for neurons. You can always assign it to single neurons because you have so much high with high resolution electrodes, that there is redundant information and there is no ambiguity between assigning signals to different neurons. You can also look at axonal arbor features, like here. That's a movie by our spin-off. They made a nice, essentially, program to do a graphical representation of an axonal arbor of a single neuron. That's the same image here, down with our means, which are a bit more modest than those of the company, obviously, software-wise. And then you can essentially extract propagation velocities along axonal arbors. So you see here, for the red segment, this is the speed, the blue segment, etc., etc. You can, with the same recording, extract many of those parameters. You see here, what you can get for axonal arbors is number of segments, for example, longest axon length, total extension, velocities, etc., etc. So all these parameters are there. And velocities, for example, are severely impacted by diseases, for example. So axonal propagation speed is clearly different in diseased neurons than in healthy ones. And the network is also something that you immediately get. You see this cross-correlation diagrams here. A is the neuron here, and you see how it is connected to the other neurons that are colored here, and here is the cross-correlation diagrams. This is a very painful method of doing pairwise correlations. In the meantime, we have much better methods to do that in bulk so that you immediately get out a big correlation diagram. Finally, I come to bidirectional interaction or stimulation. Here you have the problem that you go in with millivolt signals to stimulate and want to, at the same time, read out microvolt signals with your sensitive electronics. So 1,000-fold stronger signals are used to stimulate as you want to read out at the end. And of course, this creates problems or artifacts. And you see one of those artifacts here. If you do not do anything about that, you see that over several milliseconds, there is a huge artifact that lingers around the stimulated electrode here, which obscures any activity that there may be. So there is obviously the need to do something about it, and there's possibilities. Possibility is what we call a so-called soft reset. Soft reset is a very simple thing. It's essentially a pole shifting in the amplifiers. So what you do is you do not disconnect the electrode during the stimulation and reconnect it afterwards because they get switching artifacts. What you do is you shift the band or the high pass during the stimulation back and forth. You shift it up to higher frequency kilohertz range for a rapid discharge of the electrode, reduce the amplification at the same time, and shift it back again after the stimulation. And you see here the protocol. You start to shift it up here before you start the bidirectional or the bipolar stimulation parts, and here you see the end, here you shift it back. And that way you are within 200 microseconds back to recording. And you see it here as well. This is essentially a 1 kilohertz sine wave. If you don't do anything about it, no pole shifting, you see that it comes back after 30 milliseconds. If you do pole shifting, you see it stays in range all the time. So there is no way that you miss any signal. Here you see now the effect on the artifact that I showed you before. No soft reset, the same as before. And if you have a soft reset, you see that there is most of the electrodes they are disturbed, but they can be immediately used for recording. They don't go to the rails, and you can essentially afterward digitally subtract any artifacts or remainers of artifacts that there may be. It's very difficult, however, to recover the signal from the stimulating electrode itself, and that's why we even didn't try. Because we have so many electrodes, why would we care about this single electrode signal? Final slides here, that's a neuron. That's essentially its spontaneous activity. And you see here its stimulability. And you see that there is different electrodes, which you recognize here as well, which have different thresholds of stimulation. And you see 1, 2, 3, which are in the area of the axonal initial segment, where you get a very low threshold, 30 millivolts, whereas 7, for example, that's the soma of the cell, is about 300, so 10 times more. So the soma is particularly insensitive to stimulation because there's almost no ion channel there. And the axons are also very sensitive. You see some of those here relate to axons. Here it's better resolution. That's a spontaneous activity pattern of the same cell. Here, stimulability pattern of the same cell. You see here essentially the color coding is timing. So it arises here and then moves out in different directions. And those are signal amplitudes, the size of the blobs. Same thing here. Here is essentially the millivolts needed to stimulate in color. So dark or black values mean very low thresholds, whereas yellowish means high thresholds, and all the grays are those where even after 600 millivolt stimulation, nothing happens. So they are the insensitive region, or the insensitive areas of the neuron. And what I show you in the next slide is now a movie where we essentially stimulate at the different points here. Anti means antidromic. So it's an axon, you stimulate, the signal travels back to the soma. That's called antidromic. And orthodromic is in the right direction, so to say. It starts at the soma or at the EAS and travels in out the axons. That's the normal direction of the signal propagation. Here you see once more the thresholds. So next movie you will see exactly that situation, and we will stimulate from the different points. And we will also not use an artifact compensation so as you see where the stimulation has happened. So that's the movie. That's first the spontaneous activity of this neuron. See again this kind of flaring out of the axonal activity. It's pretty noisy because we don't have a trigger point, we don't have a stimulation point. Now you see the different stimulation points. Now we go on that side. You see that it travels this way, and then it comes out this way. And next time we'll stimulate up here, and we'll then use this axon to stimulate the neuron. So you can sit on a neuron and go to different axons and stimulate selectively this neuron through the different locations. Axons are by nature the best stimulation points simply because they are very small and can be very easily reached with an electric field. With that, I want to close and come to an end. There were two parts. I hope I have convinced you that the hanging drop networks are useful to some extent. I showed you tissue models, I showed you barrier models. And of course, we also have integrated various types of sensors, which I couldn't show today because an hour is an hour. And the second is the CMOS-based MEAs, and what do we do with those? Of course, a lot of fundamental neuroscience still. Then we also do information processing through neurons and neuromorphic structures or even circuits in the meantime. Then we use a lot of human iPSC-derived neurons nowadays, characterize those and also do disease models. And of course, as you can guess, organoids and drug screening. With that, I want to close. This is essentially the people who do our work. It's a largely interdisciplinary group comprising electrical engineers, microtechnologists, biologists, neurobiologists, patch clampers, and engineers of all kinds. We are fairly corrupt in terms that we take the money from almost any organization that wants to give money to us. Like the ERC, they gave us a lot of money, and also Marie Curie, European Union, and of course, the national funding agencies. With that, I want to close, and I'm of course happy to answer any question.
Thank you for such a delightful talk. That was remarkable. We have time for a few questions. I'm going to do this on the microphone, okay, Hung? And to everybody.
Thank you, Andreas, for coming all the way and to honor Oliver's memory. Really nice talk. I have one question. I was really taken by your two beautiful opposing way of doing science examples. I think the hanging drop is a gorgeous example of being simple and practical, and the MEA is amazing. There's all this engineering that goes into it. I'm wondering if you can comment to students or others who are interested in doing startup. What is the philosophy of choosing what's the right tool for the problem?
That's a difficult one. I mean, our philosophy is to use technology not for the sake of using technology, but for the application. And clearly, for all those microfluidic systems are driven by simplicity, by usability in incubators, by having at the least number of components possible that are not available in biology lab. So tilting stages are available, incubators are available, pumps and all this kind of microfluidic specialty stuff is not necessarily available and gets complicated if you operate many systems in parallel. Tilting, you can stack the plates even. I mean, it's very, very easy. I mean, the MEA technology is essentially our heritage from Henry's time still. I mean, it was chemical sensors, it was then MEAs. And we simply saw that in that area, there could be a lot of research done if there would be a good technology. And we then finally, 2016, also span out this Maxwell Biosystems company because many people asked us for the technology. They wanted to have setups, they wanted to have support. And our experience was giving support as an academic lab is something of the most frustrating I've ever done. It means that people take your device, put it in a drawer, think it's a turnkey system. If it's not turnkey, they will not use it. They will invest zero time and efforts into making it work. And if they don't have to pay for it, even less. So a company is the only thing to do it, and there was enough people who then bought also this stuff and wanted to have the technology. And nowadays it's fairly widespread, especially in Japan and also in industry in the smaller companies. But it still gains traction through this human iPSC-derived cell advent that really triggered it big time, because now a lot of people want to do testing or functional characterization of the iPSC cells. So there the technology was first, and then was the application that luckily came with the iPSCs. I do not think there's a strategy. I mean, it was of course delightful for us to develop all this technology, even without having a commercial purpose. But there was not a commercial driver for that at the beginning.
Thanks, Mike. Thank you, Andreas, for coming and for honoring Oliver with that amazing lecture. So maybe a addition to Hong's question. So often, because my lab deals with this quite a bit as well, you know, when you present your technology, especially to the biology side, you know, sometimes they say, this is not physiologically relevant enough. You know, it needs to be more complex. How do you deal with questions like that?
Well, actually, there is two different streams in this organ-on-chip business, or body-on-chip business, how it's called sometimes. I don't like to speak about body-on-chip because it will never be possible. I mean, you can, of course, make a fridge full of all kinds of different units that you connect, but what is the representativeness, or how much experiments can you conduct with that? Almost none. You are super complex, but the results that you get are they meaningful? And what industry wants, we collaborated from the very beginning here in those projects with Roche and Novartis. And they said, give us something simple that we can also adapt and even maybe transfer to automate or lab automation tools that we have, automated robots, plate readers, etc. All this kind of, if you don't do that, then we do not even consider it. We don't want to see it. And there we are clearly influenced by industry because we did all those projects with industry. And they told us do it as simple as possible, and they also told us what we want to see. They said, okay, we want to see metabolic effects in drug testing. That's an important thing, because liver toxicity is a major reason for drug withdrawal from the market at the moment, or toxic effects. Many of them are not even seen. And if you lose it, the drug, essentially after it has been approved, I mean, a huge investment is for nothing. So essentially, before you want to go in a clinical trial, it's extremely expensive. You want to know that your substance is not having any effects on the liver or metabolic effects. So they wanted to have that. That was really important for them. The broad brain barrier model was initially motivated by basic considerations, scientific from our side, but now we have big industry interest because they say there's nothing out there that we can use. So this is one of the systems that we really is simple enough that we think we can use it in the lab or in the research lab first, and then maybe even in the development. And essentially, they now have essentially poured money at us to go further with other models of that kind, of that simple kind. So I would say, okay, it does not reflect all eventualities, but it is reflecting what the people that collaborate with us want to see, and what they think is important for their problem or for their test scenario.
Nice talk, Andreas, thanks. The information that you are obtaining is inherently two-dimensional. That's given by the technology. Have you given any thought about the 3D propagation as it would be in a brain tissue, for example?
Yes, we did. Thank you. Essentially, that's what you developed with Nick Melosh, which is essentially a bundle of needles that you can essentially insert in the brain. And you have a 2D resolution, which is, it's actually a wire array. The wires are 5 to 10 microns thick, and they are coated with aluminum oxide and only open at the very end. And of course, this is again a 2D at a certain depth, but what you can do is essentially during the recording, you can move through tissue. And you can essentially, with a high resolution 2D array, create a pseudo 3D recording. Of course, you have the disadvantage that if you are at layer number 1 and go to layer 100, you will not be able to correlate whatever you see in layer 1 to layer 100. But you can do a layer-to-layer coordination because that's close enough. And this is a possibility. I mean, if you look at all the other things, you can ram, of course, thousands of neuropixel probes in the brain, but each of those is also 2D. And you have to still correlate those, and it's not so easy to do that. You cannot do it at neuronal resolution. So here you would have neuronal resolution, if that works. It's an idea. I don't know whether it will work. We'll see.
Okay, we have time for maybe one more question. Okay, gonna make me walk.
Thank you so much for your nice talk. I wanted to ask some detailed questions. So I don't know if you're dealing with non-specific binding or debye lengths or like say Helmholtz layer on top of the devices that you're using, the electrodes. Because when you're using these kind of electrodes for detecting biomarkers for cancer cells, you have to definitely think about these problems. If it's possible, please comment on this. And also the biocompatibility of the chip, if it's possible, please comment on that as well.
I'll start with the debye lengths, etc. first. And the debye length is also dependent on the structure size that you have to some extent. And I mean, for the neurotransmitter detection, I do not see problems with the debye lengths because essentially the neurons sit really atop the electrode array and are very, very close. They are, well, typical spacing in between is on the order of, I would say, less than a micron for sure. So there would be, if they eject neurotransmitters, you would definitely see them in a space that is close enough to the electrode. And we saw that as well. So you can see single neural events. When you see a single synapse, I would doubt that. This is probably not enough. But cells that produce neurotransmitters inject those you would see. The other thing is what you said about biocompatibility. We coat the arrays with laminin or polyalysine, and then they usually remain in the broth, so to say, for 3 to 6 months. And it means that is enough time to look at developmental processes, to look also at essentially extended times on neural processing, how it changes, even at plasticity if you would like to. However, plasticity, as we all know in vitro, is highly debated, and I certainly not advocating plasticity measurements because those are very difficult and usually not clear. They do not produce clear results. The limit in the biocompatibility is essentially by a process which is the metabolism of the cells that eat up the passivation. It was already published by Najafi, I think 30 years ago, that he could show that these typical oxide nitride stacks that are used for passivation are eaten up by neurons at a considerable speed. And the speed is on the order of 5 nanometers a day. And this is also what limit is here. So after a total cumulative culture time of 6 months roughly, those devices start to fail because the passivation is gone. Then the salt water goes into the aluminum lines or copper lines, dissolves them, and then chips are dead with a couple of bubbles that you see, and then it's done. That is a limit there. You could, of course, make this passivation very thick, but usually it doesn't help much.
Great. Thank you, Andreas. Let's give him a big round of applause. Two things, just a reminder that there will be a reception immediately following in the atrium. We hope you all be able to join us. Before we step out, want to give Andreas a token of our appreciation for traveling all the way from ETH Zurich. Sorry for the rustling earlier. This is a beautiful plaque fabricated right here in our Georgia Tech clean room, showing the seal of Georgia Tech. It says, Andreas Hierlemann, distinguished speaker for the first Oliver Brand lectureship on electronics and nanotechnology. We'll see you outside.